Okay, the last time we talked about protein purification and also case study. So now that is the topic is move on to the mammalian cell now. The mammalian cell, how we grow, how we culture the mammalian cell. The reason is actually we have to in the curriculum of the chemistry department in KAIST. Actually, we don't teach how to grow the cells. But it's actually very critical if you think about these days that the organic chemistry, for example, that you developed some potential drug, for example, like a COVID-19 drug you synthesize. And then the next step is you have to check about the efficiency and safety and that cytotoxicity and everything. So then you have to basically, the system goes into the mammalian cell culture. You've got a drugs and then cell, and you put it into the cell and how they die, how much the cells are dying, or proliferating or differentiating kind of things. So it's very important to know to understand the chemical biology and then know the mammalian cell, how they grow and the principle, what is the basic principles, also mechanisms, molecular levels and simple things, not in deep level of the cell biology class, but actually has to know the essential part that you have to know. Okay. So now begin. Then the mammalian cell culture. So mammalian cells, we have to talk about size first. So then I'll draw like this. Or draw like, say, like this. Maybe you can imagine this. What is this? What is that? So this is a bacteria. For example, E. coli. So this bacteria that they also can by themselves are divided. So the bacteria, for example, is the molar size is about 1.5 microns or 2 microns. So it's depending on there's a distributions on it. So actually draw in a right scale. So if this is a bacteria, then 1, 2, 3, 4, 5, 6, 7, 8, 9, about 9. Then it's more than 10. So I should draw like even bigger than about like this. About like this. So this is actually the size of the mammalian cell. So more than 10 microns. So then this is from here. From here is bigger than, say, 15 micron. That is like a 10 times difference. Of course, the height is actually, I should draw more thicker than this, like this. So then if you can imagine, if this is the overall size of the mammalian cell, that our cell in our body, then the bacteria is about this size, or even smaller that goes in there. Okay, this kind of things. For example, muscle, that in muscle cells right here, or heart also. So cells begin to fuse to each other. So one cells, the other cells are another cells you can imagine. Now their membrane, which is membrane like this, is they fuse to have another cells goes in here. So that's why you have a muscle fibers, is fibrous things that actually you can see multinucleus. So the cell has one membrane there, whereas multi-nuclei are there. So they fuse to each other, so it becomes fiber. But that is kind of muscles that how they grow it. So anyway, the basic scales of the size is like this. I will delete this now. The basic thing then we have to know is the mammalian cells are mostly the anchorage dependent cell. Anchorage dependent means that the cells has to stick on surfaces. So they have stick. In other words, the cell has to attach on a surface. So that means, for example, it means if you bring a piece of glass, and you got mammalian cells plus a mammalian cell, say mammalian cell, then the cell goes to the surface of the glass, they can grow. So basically growth condition. But it's not, but it has to be anchorage dependent. That means attach to surface. So if you bring a glass and a cell, that has to grow. But actually this grow is not a right answer, because there's another factors. Another factor is most important thing, which is a protein always a protein. I should say proteins are required. So in other words, that now we combine all this. So proteins are required, cells are anchorage dependent, so they has to stick on surfaces. So that means you combine this, then require a surface that to which proteins are absorbed. So this is how we saw the first things that I want to teach you, that to grow mammalian cells, most anchorage dependent cells, fibroblast, myoblast, neurons, hepatocyte, which is a liver cells, many different things that has to be surfaces, anchor on surfaces. But if you bring, for example, the piece of glasses or this surfaces, and you got a cells in a solution, you put the cells, they didn't grow, because has the proteins are required. So now you can think that you got a surfaces, and to which the proteins has to be existed on surfaces, and then the cells recognize this protein and bind. So then they grow. So that is kind of principle. So it's very simple, but easy to forget. Surfaces that require a protein absorbed surfaces. So that is one thing. It's very, very important things that have to understand. So the reason is the cells has a lot of membrane proteins. For example, remember that COVID-19 class that you got a draw like alveoli, right? And then you got a cells here. For example, like this. Oh, I'm not going to draw all of them. There are the cells in alveoli. So remember that there's a protein actually I called ACE2. Remember that one is angiotensin converting enzyme 2, right? So that is where the protein exists. The protein exists in a membrane that is a transmembrane proteins that I explained. So then the COVID-19, this coronavirus, lines here, they go with this is entry mechanism. It's the first entry mechanism is this one. So in other words, the mammalian cell has always have the membrane proteins. A lot of different membrane proteins. Like generally speaking, for example, integrin is the name of the protein that recognizes the extracellular matrix we call the ECM. So extracellular cells producing protein for our environment. That is called extracellular matrix that we call collagen. So one of the representative ECM proteins is collagen. Maybe elastin. So collagen and elastin are famous because they're cosmetic product related. So there's this collagen and elastic so does not exist inside cell, rather exists in outside cell. So that's why extracellular matrix. That integrin recognize it. Integrin is a membrane protein. Integrin is membrane that as a cell has. This is the ECM. Then this is a cell. This is the integrin that recognize it. That's why cells require surfaces on top. The proteins are absorbed on top of it. So that is the most important single principle that for the mammalian cell most cells require surfaces, but surface has the protein on top. So that is one thing. Okay. So then now remember the size. So now remember the size of the protein. That's why we begin with this class is about teaching you about the size of the macromolecule, size of protein. It's about, say, now remember that, say, about this, maybe 2 nanometer, 3 nanometer, 4 nanometer, something like this. Right? If you don't know it, then you have to go back to the macromolecular size classes, part 1, part 2. Which is very, very important. So now, then you think about it. Your surface is here. Say you got a glass, right? Glass, and then if this is a layer of the protein, for example, from this here, I'll do a different. Say from here to here, we have a layer of protein. So then I will ask you about what is the thickness of this layer. So say that you have a glasses with a protein on top, with a glasses of protein without protein on top. So no protein and protein. Can you distinguish? No, because you have a layer of protein, the thickness is just 2 or 3, 4 nanometers or 10 nanometers. Then how your eyes is such a good to distinguish between the transparent glasses with the protein with the thickness about 3 nanometers, 4 nanometers. You basically cannot distinguish. So that's why you can easily tell mammalian cells grow on the surfaces, but it's not true because as you cannot actually distinguish by your eye, cannot see the protein layers. Okay? It has to be activated. A surfaces that has a protein on top of it. Okay. That is one thing. So the protein now, so then the protein is, then what kind of protein we use? That's the next question. So proteins we use is the serum. It's from the serum. What is the serum? It's a blood protein, right? This is a blood protein. So where does the blood comes? Blood comes is, say, cough. So because we eat cow, right, beef, then you have a lot of blood. So you gotta so we get a bovine, okay, and especially when we the source is actually when they have a pregnancy, then they be baby cow, then is a fetal, which is fetal part that you have a connection between the mother and this daughter. That part is out bleeding, and then they have collecting those things because they can throw that away. Because you cannot throw that away. Mother cannot. Their daughters cannot throw that away, right? They have to grow. But the fetal part, they have to just trash. You can just throw that away. So then fetal bovine serum is the source. This is a very cheap source, a large amount, and then you take the protein, and then you have a certain very controlled, carefully controlled procedure. Then you got proteins from the blood. You got serum proteins from here. So you got solutions of the serum protein. I will show you the next in a moment, that in a slide, that the serum looks like a yellow, and then you put say a piece of glass. You prepare the serum solution. Glass go in here, maybe 5 to 10 minutes, depending on maybe 1 hour. Then proteins goes absorb on top of the glass. You take it out. Serum, you cannot distinguish, by the way, it's still very transparent. A few nanometers in thickness. Then you can now cells can recognize and they can grow. Okay. So the previous previously I mentioned about the fetal bovine serum, which is actually how it look like. So the fetal bovine serum is here. So this unit is about 500 milliliters they sell. You see this is yellow color. So a lot of proteins are here. So basically you have these solutions. Essentially you have to buy it, and then we mix with this so-called mammalian culture cell media. We mix with this. Also, if you have a proteins, the surfaces that has this serum proteins absorbed, and then bring into the serum-free media. Serum-free media, so there is no serum proteins, but there is here. Then there's probably a certain period of time, the mammalian cells actually can attach, and they anchors, and they spread it and then proliferate it. So this is the how they look like. Then this slide is basically give you the culture cell line, called the cell line that is it's a long time to explain, but it's short in brief. That is name of the cells are representing the cell type of fibroblast or epithelial, lymphoblast, and also probably that, but actually source is say kidney. You can see a very different some carcinoma, marrow, lung, for example, embryo, breast, liver, something like this. So that means liver. You have a liver, and epithelium means it's the outside of the liver. So that is called the chung cell. Basically, general cell that you take the cells directly from the liver. We call the primary cell, and primary cells are you can actually culture this serum-containing media, but they don't grow and divide it well. So this cell line means that this is the forever they actually can divide it. So that means there's some cancer type of the cell that can kind of fusions and clone, that we especially got a clone on this cell, and then the cells we collected maintaining this, for example, kidney fibroblast, but that they have maybe 95% of the kidney fibroblast, but they plus a little bit of the cancer cell type they can they can continuously grow. So it can easy to culture this cell line. So that's kind of concept of cell line. So the important thing is in media, where they grow. So then MEM and DMEM, F12K, this is the compositions of the, for example, buffer and vitamin, pH, and glucose, those kind of things. Plus FBS is I explain about fetal bovine serum. Say 10% in a volume. Say you have 100 milliliters, then you must have a 90 milliliters of this and 10 milliliters of the FBS you have to have. Say sometimes you have a 15, sometimes in 20%, for example, depending on. So then, but typically you can see 10% of the FBS, right? Most are 10% FBS. See also 10%. Sometimes it's 20%. Say barely 5%, but it's not many cases. This is kind of concentrations is needed. But the thing is, there is no culture condition or there's no medium that without serum FBS. There's nothing, because the serum is very essential in growing the mammalian cells, right? And also, so I will show you the media compositions, in general compositions of the media. We call this DMEM or MEM, MEM. The formulation is basically some salt, inorganic salt, sodium salt and potassium salt, calciums, and also amino acid of the 20 different amino acid or essential amino acid we have to add it, and also the vitamins that is shown here, and others such as glucose, phenolate, and sodium pyruvate. I will explain not one by one. This is quite in detail why they need it. Right? The plus, what is it? Plus 10% FBS, which is a protein that we mentioned. So among this, what is the most abundant amount that we added? So basically, this sodium chloride, right? Sodium chloride, and also the glucose is the most important things, because there's a lot of amount is added to it. Sodium chloride, osmosis pressure, like matching the salt level between outside and inside. The osmosis control. We need some calcium. We need some iron. We need some magnesium. We need some potassium. And this one and this two, sodium carbonate, monosodium carbonate, monosodium phosphate buffers. This is a buffering things of matching the pH, or maybe not. pH is deviate from the 7.4, so you can buffering of this overall system. And then for protein synthesis, you need amino acid, and also other vitamins, and folic acid is essential. Riboflavins and all the enzymatic metabolisms inside the cell. And the glucose is energy source. Remember the TCA cycles and sort of things. You can get an energy source, become the glucose, and after TCA cycle, you got ATP, and this is one metabolism class that you already learned in biochemistry class. The phenol red is the indicator, it's a pH indicator. So that is generally at the neutral pH, they exhibit red color. But the thing is, when cells are growing, 1 becomes a 2, 2 becomes 4, growing in the situations, they generating basically acid, right? So that means that they got a glucose, but becomes lactic acid, for example, after making ATP, or many other acids. So they become oxidated and then become acid. So the overall pH of the system of the when you grow the mammalian cells drops down. So when this how you know it, without this pH indicator you cannot see it. But with this pH pH indicator becomes orange color. So now we can see this is time for changing the media by you actually see the color of the solution becomes red and orange. Orange need to be changed. So something like this. It's sort of like a very complicated, but this now we can understand quite a good levels of how they meet compositions of this media. So this is how they look like. So you sit in here, right? You sit in here, and then this is the T70 culture dish, in which inside you got this is media. So previously this solution means is DMEM, which is this one, and then you plus 10%, for example, FP bovine syrups. This is the solution for it. And then where is the cell? Cell is on top, attaching to on a bottom surfaces of this culture dish. So you bring this to the 37-degree incubators and goes in. They grow there. For the sterilization, 70% ethanol. So when you go sitting there and then you take this and then you spray and then you got all sterilizations, also inside and outside here. Okay, so this tubing, because you when you change media, you use the pipette, and then there's a pipette, say 10 milliliters pipette. You take all the media out of this glass, and then you got a new media goes in there. Okay, something like this. And this is a waste. You have to throw that away here. And then you got a certain other pipettes, not seen, but there's a pipette in here. So you got pipettes and goes in, and then you take this and goes in here. It's simple. So then is after this, I can explain one by one about how they actually what kind of procedures of the growing the mammalian cells. Okay. So for now, this is the end of the class. So next one we talk about the step-by-step protocols how we get growing of the mammalian cell. Okay. Thank you very much.